Very good evening, everyone. I'm Dr. Himani, and today, on the behalf of Vital Biotech, Agriculture Forum for Technical Education of Farming Society, Kota, Faculty of Veterinary Sciences and Animal Husbandry, Ranchi, Birsa Agriculture University, Ranchi, Jharkhand, I cordially welcome you all to the fourth day of this international training come workshop on cutting edge innovation in veterinary science for a sustainable future. Before starting, let me tell you a little bit about the organizers. Organizers are Vital Biotech. It is India's leading publication, research, and training institute, whose goal is to tackle the world's most pressing problem, which are related to food and agriculture. Whereas the Agriculture Forum for Technical Education of Farming Societies, it serves as a spiral between the network of rural communities in India and other stakeholders such as government, developmental organization, and corporate entities. And when talking about Birsa Agriculture University, it is a well-known name. It was established in 1981 and inaugurated by then Prime Minister late Srimati Indira Gandhi ji. Primary objective of this university is to develop area-specific technologies and manpower in the field of agriculture, animal husbandry, and forestry for the agriculture development of Jharkhand and economic upliftment of the tribal and other backward classes of that area. And research, teaching, and extension activities are being carried out there within 7 faculties and 12 colleges. And beside this, university has another College of Biotechnology, Center for Agribusiness Management, 6 directorates, 3 zonal research station, university farm, 16 KVKs, computer center, central library. So the university is well equipped with all the infrastructure and service units. And the College of Veterinary Science and Animal Husbandry, it was established in 1961. Initially, it was a part of Ranchi University. Later on, it became a constituent of Rajendra Agriculture University. And then it focused on UG programs, but then PG programs were added in 1972 and it was expanded. In 1981, it was incorporated into Birsa Agriculture University, and since then it is offering UG, PG, PhD diploma and other certificate courses. So the college is involved in research related to animal health and nutrition and conduct extension activities like livestock health camps and farmer education. So once again, I want to congratulate the entire organizing team and organizers for organizing this insightful training program in which we are getting a lot of response from the participants. So dear participants, moving forward, today we are highly privileged. We have an honorable dignitary of today's session with us. So it is a matter of great pride that today we have Dr. Nirbhaya Kumar, who is professor and head department of veterinary pharmacology and toxicology at Bihar Veterinary College, Bihar Animal Science University, Patna. So Dr. Kumar is a distinguished academic and academician and researcher. He is currently serving in the Department of Veterinary Pharmacology, and sir has done his BSc in Veterinary Sciences and Animal Husbandry, Masters in Veterinary Sciences from Bihar Veterinary College, Patna, and sir holds a PhD in Veterinary Pharmacology and Toxicology from GB Pant University of Agriculture Sciences and Technology, Pantnagar. With over 20 years of experience in teaching and research, sir has served in various capacities, including assistant professor, associate professor, and now professor and head. Sir is principal investigator of the ICR-funded network project, which is AINP on All India Network Program on Livestock and Poultry Product Safety. His academic contributions include mentoring 1 doctoral and 8 PG students, publishing over 50 research papers in esteemed national and international journal, authoring 2 books along with 27 book chapters. Sir has delivered numerous lectures at conferences, seminars, and training programs, and has additionally organized various training sessions to build research and academic capacity, and he has been actively involved in the extension activities and ultimately benefiting the farmers of his state, Bihar. The outreach of Dr. Kumar efforts include delivering 7 television talks aimed at educating the farmers and the agriculture and veterinary community. Sir, your achievement shows us that you are indeed a source of inspiration for all of us, and the participants will truly learn a lot from you today. So today, sir is going to talk on the topic, which is utilization of artificial intelligence in drug discovery and delivery within veterinary sciences. So, Sir, now this virtual stage is yours, and you can just start with your talk. Over to you, sir.
Thank you, Dr. Himani. I'm sharing my slides.
Yes, sir.
Is it visible now?
Yeah, sir, perfectly visible.
Yes,
Okay.
sir.
Okay. So, very good evening to all of you. And first of all, I'd like to thank the organizers for giving this opportunity to share my ideas over this very beautiful and new topic, an innovative topic, in fact. Well, I'd like to thank Dr. Dinesh also, who personally interacted with me to have a talk on this topic. And I'd like to thank the society as well as the Birsa Agriculture University, from where I'm having interaction since so many years. To start with this topic, we all are using artificial intelligence in so many fields in day-to-day interactions. And the uses of this artificial intelligence in veterinary sciences, especially in drug discovery, in discovery of new drugs, and how they should be delivered. So in drug delivery. Now, it's not a new topic in veterinary pharmacology, because from 1970, 80s, these techniques were used, which we call them as computer-aided drug discovery. And now, with addition of new technology, we are moving forward. So to start this topic, I'll be discussing with very basics, and then we'll expand till the developments in science. So because I think some students may be also there. So to start this topic, first let's define what is artificial intelligence. So, as this term says, it refers to the simulation of human intelligence in machines to perform tasks such as learning, reasoning, and problem solving. We know that artificial intelligence, it is not exactly the human brain, but how we think, how we correlate data, how we recognize something. So that's why it's called artificial intelligence, because it is the simulation of human intelligence with the help of some machines, so that we can perform some defined tasks. Now, nowadays, there are so many types of artificial intelligence. One is machine learning. We'll discuss later on one by one all these things. Second is deep learning. Third is natural language processing, NLP. And the last one is computer vision. So let's define all these things one by one, so that we can understand the topic in depth. So what's this machine learning is all about? It is a type or it is a subset of artificial intelligence that enables computers to learn from data and improve their performance without being explicitly programmed. So if we submit some data to the computer system, the computer can make its own analysis and can analyze the data, and can come up with some summary or conclusions like that. So based on certain filters, if the machine is learning or machine is concluding something, we'll say it as machine learning. And how it works, let's understand. So machine learning algorithms analyze data, identify patterns if there is any pattern in the data, and make predictions or decisions based on those patterns. So this is the technique the machine uses. And we can have some examples, like we have some spam emails in our mailbox. So it identifies and filters out unwanted emails which are not being routinely or which are not part of our directory. So this is one example. It can be the machine or the system recommends us, so suggesting products or content based on your preference. So if you are using say for example, nowadays we listen so many songs, and the first song whatever you listen, so based on the taste of that song, the system automatically says second, third, fourth songs. So this kind of learning is machine learning. The machine is learning your taste and recommended other so many other songs so that you can. So the second component of AI is deep learning. How it's different? Let's understand it. So deep learning is a subset of, or it is a subfield of machine learning that uses artificial neural networks with multiple layers. We have made some neural framework into the machine learning, which we call it as deep language learning. And deep neural networks can analyze data. So how it works? So deep learning algorithms can automatically learn hierarchical representations of data. If we are seeing so many complex data, so it can make its own hierarchy level 1, 2, 3 like that, and which can allow them to identify complex patterns and make accurate predictions. So let's see how it works and what can be the examples. One very most important and day-to-day example is image recognition in Google Images or in Google Lens. If you fit any image, it will search from the directory and it will identify what this object is all about. So identifying objects in images like faces or cars, we use in our mobile also face recognition. So not only that, speech recognition. So your voice, so many nowadays systems are mobile or your entrance lock of your things. So some human neural definitions have been incorporated so that it can recognize image voice like that. The next part is natural language processing. Nowadays, we understand our own language. Someone speaks English or Hindi or Marathi and all these things. So understanding human language and translating them into so many other languages is very important. So it focuses on enabling computers to understand, interpret, and generate human language. So how it works? Natural language processing (NLP) uses machine learning and deep learning techniques to process and analyze text and speech data. And so many examples are there. There are chatbots, AI-powered virtual assistants that can understand and respond to user queries. If you visit any website, you will find some chatbot or AI assistant. And if you click and if you say in your voice or if you type any query, it will filter out and it will come up with some suggestions or replies. So those are the chatbots. There are machine translation you will find in every website, bilingual or so website designed in so many languages. So the texts are being translated to another languages. The next and the last type is computer vision. So what we want that whatever we think, whatever we execute, the machine or the computer should do on our behalf. So exactly, the system cannot replace human brain, but we can make it to think like ours. And the latest addition is computer vision. So computer vision is a field of AI that enables computers to see and interpret images and videos. And how it works, computer vision uses algorithms to analyze visual data, identify objects, and perform tasks like image recognition and object detection. And the examples are in so many countries, you will find the self-driving cars. The car is being connected with the internet and is being given geolocation and the directions. And based on those directions, the system is running. So using computer vision to perceive and navigate the environment. And medical image analysis. Nowadays, we see ultrasound, we see X-rays or the MRI scans or the CT scans, and the doctor is analyzing based on the image pattern and all these things. So we can learn the computer. We can tell the computer. We can train the computer about the different kind of images. Say, for example, histopathology images, we can tell the computer if this is the pattern, this is the normal liver tissue. If this is the deviation, it can tell us the carcinoma or if the hepatic cancer or like that. So based on the image, the system can automatically suggest us or can come up with some solution or the conclusion that this image is correlated with this. So identifying diseases or abnormalities in medical images, medical imaging field nowadays is very broad, and so many techniques are there, like X-rays or MRI scans or computer topography and all these things. So this system or the computer vision can help us. So nowadays there are so many deviations of these, and there are so many applications of these AI techniques in medical sciences, especially in our veterinary science. So let's see how it can help us and how it's helping. We all are using so many veterinary drugs and formulations for treatment of our animals, but we may not be knowing that it may have been developed with the help of some AI techniques. So how this AI in veterinary science can help us? It can help us in faster and more accurate diagnosis, in diagnosis of X-rays or in diagnosis of histopathological slides or in diagnosis of CT scans and all these things. AI-powered drug discovery and development. Nowadays, we find that there are so many disease conditions. Recently, we faced COVID, and till date, we don't have any sure shot medication for treatment of this disease. And there is global pressure over scientists for drug discovery and development, that new drugs should come up in the market to cure these challenging disease conditions. And in drug discovery and development, because this drug discovery is very complex, sophisticated process, involves 4 stages, which may cover up to 10, 12 years. So if we can use artificial intelligence in different stages of drug discovery and development, we'll discuss later on all these things. And definitely it can reduce this 10, 12 years life cycle. And more number of newer drugs may come up in the market with the help of these techniques. And precision medicine and personalized treatments. Nowadays, because a single drug is being used in the whole human population at the same dose, myself or some other body person is receiving paracetamol at the dose rate of 5 milligram per kg body weight. And all humans or all individuals are not exactly the same. They may differ in their genetic makeup. They may differ in their drug behavior and responses. So there is a concept of personalized medicine, that each and every individual should be given individualized medicine, personalized medicine based on their genetic makeup or based on their individual physiology. Grossly, physiology is same throughout the human population, but there may be differences in drug metabolizing pattern of any particular drug. I may differ with myself with some others. So in the selection of this personalized and precision medicine, this AI can definitely help us. For this treatment, just to share the background, United States had made a very ambitious project of whole human genome sequencing across the globe. Because there are so many countries, there are so many geographical locations. Some Chinese look different, Thai people look different, Negroes look different. Throughout the globe, every human population was sequenced. Their genetic makeup was sequenced with the help of real-time PCRs and all these sequencing techniques. And the whole genome was explored. And after that, because making the whole genome throughout the globe sequenced and each and every gene to be identified, it's not a simple job. It's a very sophisticated job. And now there is concept that after some years, say for example, I may have a card, or like our Aadhaar card, I may have a card that in which my whole genome sequence may be mapped. And once you visit any doctor, you may give your card number or your code, and from the cloud directly, he may see that if I am suggesting paracetamol to you, whether you are paracetamol sensitive, whether your body will behave differently to this drug. So based on your personal genetic makeup, the doctor can be able to recommend you your personalized medicines. So this AI, artificial intelligence, can definitely help us to develop this personalized medicine treatment. Not only that, automated monitoring of animal health, because in veterinary science, we are having farms of cattle, buffalo, 500,000 cattle, buffaloes, and one or two or three veterinarians are taking care of their health. Now, to check each and every animal individually every day is practically not possible, because we have limited vets appointed at the farm. Now, what's the requirement? Requirement is that each and every animal should be checked individually, so that if someone gets sick, it can be segregated out of the herd. So, how we can take care of this? With the help of AI techniques, we can fix some chips over ear, or make some tags, and we can monitor each and every animal for their body temperature in a system, and the AI can uh, make you sensors and can make you some suggestions that animal number 736 is having fever, or so. With the help of these techniques, the automated monitoring of animal health can be performed. So obviously, this AI is a new technique, and it's having some advantages, but it's having some challenges also. So artificial intelligence versus traditional medicines in methods in drug discovery. Let's compare, because my assigned topic is drug discovery and drug delivery, the applications of artificial intelligence. So uh, let's understand how drug discovery is being made traditionally. As I told you that this drug discovery is a complex process which covers 10 to 12 years. Let's take the example of this COVID vaccine. When phase one infections came, after the phase two, after having a lot of mortality, each and every country throughout the globe, each and every scientist was engaged in development of corona vaccine. And after 2 years, so many countries came up with their own vaccines, but they are not open to use. You cannot purchase any COVID vaccine directly from the shopkeeper, because this drug discovery process is 10 to 12 years. Only after completion of this period, the drug can be made available in the market freely. So each and every government gave emergency approval of the vaccine, because the human life was going out, and that's why it's emergency approval, and only phase one and phase two are cleared. There are phase three and phase four. It's not completed. What's these, just to have some brief data in different phases. Once a new molecule is developed, once a new drug or COVID vaccine is developed by some team of scientists or by some personnel, it has to be tested for its safety first, on safety on so many parameters, whether it's having any toxicity for our GI tract, whether it's having any toxicity for our respiratory system, whether it's having any toxicity for our kidneys, for liver, for lungs, for our reproductive systems. So all these safety studies are being performed. It's more than 6 months process, and you cannot test them directly into the human beings. First, it has to be done in some cell lines in vitro models. Then they'll be replicated in small laboratory animals like rat, mice. Then later on to sheep, goat, and then to dogs. And then based on these findings, if everything goes well, no any untoward actions are being there, and these preclinical, we call them as preclinical studies. Once the preclinical studies are being completed, then it's ready for human clinical challenge. And then defined volunteers are being selected. And you might have, you can recall that once the COVID vaccine came into the market, some very high profile hospitals in India were selected, and volunteers were identified that I am voluntarily giving myself or offering myself for treatment of for challenge of this vaccine, for efficacy study of this vaccine. And and that's being done only on small scale, on some 40-50 individuals. Based on the data, based on the response of 50 persons, another 500 persons are in different locations in the country are being exposed. And based on those informations, so it is slow and gradual process. You cannot expose the whole human population all of a sudden with any new drug. If it becomes toxic, if there is a large human mortality, you cannot do it legally. So how that this our artificial intelligence can help us in traditional methods in drug discovery. So drug discovery is a complex process that I have told you. It's a 10-12 years process involving so many stages that traditionally, but AI is revolutionizing this process, such as speed fast in research and improving accuracy. So it can definitely reduce some of the stages. And what are the factors involved? Factors like time efficiency, cost effectiveness, accuracy, and predictive power, data handling and analysis, and failure rates. It can take care. It has to take care of all these factors so that traditional methods in drug discovery can be analyzed. Let's compare the comparative analysis of artificial intelligence and drug discovery process. In traditional methods, as I told you that normally 10 to 15 years time is taken for any new drug discovery. And if we use AI powered methods, we can reduce it to 1 to 3 years. And if we can see the cost in traditional methods, the cost is very high in billions for development of one new molecule and launching to the market. And with the help of AI methods, definitely we can reduce the cost significantly. And what about accuracy? In traditional methods, accuracy is limited because human calculations, human involvement is there. So accuracy is limited. But in AI-powered methods, with the help of computers, it is high predictive power and accuracy is more. If we see the data analysis, because analyzing millions, trillions of data manually, it's time consuming. It will take great time. But if we use AI powered methods, it is automated and rapid analysis, and failure rates are also very lesser compared to more than 90% failure rates in traditional methods. So this is the power of AI, and this is the advantage of AI over traditional methods. So what are the advantages? There is faster drug identification and development. There is cost reduction. There is more precise predictions. There is better handling of big data, and there is improved safety through early toxicity detection. So these are the advantages, and obviously there are challenges because machine cannot replace human brain. So this is the major challenge, and high initial implementation cost may be there at the initial stage, because we have to design the system in such a way that it can take care of all our manual efforts. So need for high quality training data, and the personnel involved in analysis, they can be trained. And obviously there is regulatory and ethical concerns are there, because if the machine is making some prescription based on AI, whether it can be challenged in high court or in law, whether it's perfect prescription, because a prescription written by a registered medical practitioner or registered veterinary practitioner is a legal document. It can be challenged in court. If the doctor is writing some absurd, he or she can be punished. But what about the computer prescription by AI? So it is a regulatory and ethical concern. So these concerns obviously cannot be replaced. Human brain cannot be fully replaced. We can take some advantages of the AI and lack of interpretability in AI decision making based on because the doctor is taking all your symptoms and knowing these, and after that the doctor makes some diagnosis that you are suffering from this particular disease and then makes the prescription. So the AI-enabled systems can make some decision, but the interpretability of those decisions in AI decision-making models are not fully replaced in medical sciences by which human brain was doing. So that's another limitation. Obviously, there are so many fields in which AI is doing wonderful, and one important field is nanotechnology-based drug delivery. You all may be knowing that this nanotechnology is a new development in science, in which we are making everything in nano scale. Nano is 10 to the power minus 9. So nano, if we make any particular thing very small, what can be the advantages? Now, in normal drug delivery, say for example, I'll take you one example. If you are giving paracetamol, if you are giving any particular drug, if you are giving any particular drug to the human body, say for example, this drug is paracetamol, and the dose, what you are giving 500 milligram tablet. You are giving 500 milligram tablet. Where it goes into the stomach, where you are taking with 1 glass of water. It will dissolve in water, and all 500 milligram may not be dissolved. Some people takes only 1 or 2 sip of water. So whatever drug is being dissolved, only it will be absorbed. From here, it will be absorbed into the blood. Undissolved part will pass into the feces. So, say for example, 400 milligram was dissolved, and 100 milligram was undissolved. So, 100 milligram was passed into the feces along with your stool. So, this is the first loss. The dissolved part will pass here and will go first go into the blood, and from blood it will go to the liver. Liver is the boss of all the organs. Whatever you are taking through food, it will go to the liver, and liver is having so many enzymes. It will metabolize. So it may metabolize 20% of the drug. So it is and the unmetabolized may come up in the systemic circulation. So, for example, 320 milligram is coming to the systemic circulation, and 80 milligram was destroyed by the liver. So this is the second loss. In the blood, there may be so many enzymes which may metabolize this particular. Say, for example, some 20 milligram was destroyed, and the remaining 300 is circulating to the blood. And what is the target of this drug in the body? There may be some target where this drug will bind. Once this drug binds to its target, goes to the target, what is the required amount? Required amount of required concentration responsible for activating this target may be very, very less. Some 5 milligram, 10 milligram total may have been was sufficient, because this is traditional route of drug administration. If your receptor is located somewhere, you are giving through oral route, and it is passing all these stages. So this is traditional drug discovery process. This is traditional drug delivery process. And based on nanotechnology, we are making this 500 dose to some 5 milligram dose or 1 milligram dose, and we are encapsulating this nanomedicine in some nano vehicle, and we are giving in such a that it will be released only at the target, nowhere else. So whatever amount, whatever drug we are giving, it will be delivered to the receptor or the target directly without any alteration. So what happens that initially in normal drug delivery, you was giving 500 milligram of the drug. So the toxicity of any drug or side effect of any drug is directly proportional with its dose. The side effect or the toxicity was directly proportional with the dose. So you are taking 500 milligrams. So your liver, because this paracetamol is hepatotoxic, it damages your liver. So damage of liver was to the concentration of 500 milligram. Once you are giving a nano medicine, you are taking only 500 milligram. So the toxicity is reduced 100 times because only 500 milligram is there in the body. So one advantage of nanotechnology is reduction in dose. Reduction in dose is directly proportional to its toxicity. So there is reduction in toxicity or the safety. Safety is increased. So this nanotechnology-based drug delivery, definitely it has some component where we can use AI. So it optimizes nano particles to improve drug absorption, targets specific tissues with greater precision, and the benefits reduces side effects, enhances drug stability, increases bioavailability or the availability of the drug in the systemic circulation. And the example can be AI-enhanced nanoparticles for cancer treatment in dogs. For treatment of cancer, now this is such a disease that till date we can say that it is some type of incurable disease. It is not fully cured. Either you make surgical incision and you remove the surgery, remove the affected part, you give irradiation or you give chemotherapy. After that, also the disease is not fully eradicated. So globally, a lot of research is being going on for treatment of disease, and the AI is definitely helping to develop new novel molecules for novel treatment. Now, there can be some smart drug delivery devices also. So AI-driven implants devices that automatically adjust drug delivery. We can make some implants over the skin, just below the skin, or make some, say for example, some intrauterine implants or some other implants also. And we can fine tune, we can adjust the device in such a way that it will deliver 3 molecules every day to the systemic circulation. So obviously it is controlled release. Examples can be smart insulin pumps for diabetic animals. If animal or the human being is diabetic, now what's the treatment? Every day before food, the patient takes one intraperitoneal injection. It's very painful because खाने के पहले आप एक injection लेना है. But if we can make some implants, automatically it will continuously release insulin, and it can be helpful. AI-controlled drug patches for long-term medication. If any drug addict is there, कोई drug addict है जो गांजा और कुछ लेने का उसको every day it's having some habit. So in treatment, we can make nicotine implants or some implants beneath the skin, which will release slowly and slowly and slowly, and it can help in the recovery of the patient. So implant releasing antibiotics for cattle also. So many applications can be developed. So emerging AI trends in veterinary drug discovery, AI-powered precision medicine, personalized treatments based on genetics and health data, they are being evolved. AI and quantum computing for drug discovery. So faster molecule simulations for new drugs. AI-driven high throughput screening, automated AI testing for thousands of compounds. AI in herbal and alternative medicine, AI in studying natural compounds for animal health, AI in predictive pharmacology, AI modeling how drugs will react will react before testing. So all these fields are AI is being used in all these fields for development of any particular new drug. Nowadays, Ayurvedic medications, so many people are using, and there is a lot of research going on in Ayurveda. And in the Ayurvedic medical system of medicine, most of the drugs are comes from plant origin, and which particular drug is having which particular compound is present in more concentration in particular Ayurvedic medicine. So, based on AI techniques, based in predictive pharmacology or in alternate herbal and alternative medicine, these techniques are being used and they are helping a lot for development of new drug molecules from these systems of medicine. So, the role of big data and AI in veterinary research. Once you go for research or development of any new molecule, you may have very big data to handle. And say, for example, the data of whole genome information of human being throughout the globe. How it can be handled? It requires information of bioinformatics and those softwares which can analyze the data. And obviously, if we can tag these bioinformatics with some AI tools, it can sum up with it can come up with its own conclusions or recommendations. So how big data is changing drug discovery and delivery, millions of veterinary records for drug trends, and integrates genetic, environmental, and behavioral data, predict how diseases evolve in animals, and examples like zoonotic disease outbreaks. So this COVID is also a zoonotic disease. It has come up from the animals. It was originated in animals, and now it's affecting human beings and vice versa. So for data handling and for management of these diseases in veterinary as well as medical side, this AI is helping a lot. AI and robotics in veterinary medicine. It's a new field, and how it can help us perform precision drug injections. If we can make some robots in as our veterinary assistants, as our assistant to any veterinary doctor, and the robot can make precise, accurate drug injection, like intramuscular injection or intravenous or subcutaneous injections. Automated diagnostic machines for real-time results. We can make some robots, robotic systems, and we can make AI informatics into the robot, and it can make diagnosis. It can scan your body. It can make MRI scans, and it can automatically. We can reduce so many steps. So autonomous vet drones delivering medicine in rural areas. We can help take off drones, and those drones can be tagged with AI techniques, and those drones can identify based on from distant location, can identify the temperature of the animal in the some rural field or in the jungle or in some this forest conditions, and based on that it can tag. So there can be so many applications where directly human beings cannot go, and we can treat them with the help of these robotics. Examples can be robotic surgery for complex veterinary procedures, and so many can be. So AI-driven drug development for emerging diseases. AI can predict and combat new veterinary diseases, potential animal pandemics early, rapid response vaccines, new antiviral drugs in virtual simulations. And the examples can be faster vaccine for avian influenza. Avian influenza before COVID was a big pandemic, big concern. Obviously, there are ethical and regulatory challenges in AI because it is up to what level it can be accepted. So that's an issue. So ethical concerns are there. Transparency in AI, the AI decision making is being challenged. Regulatory hurdles are there. If approving AI discovered drugs for veterinary use or for human use, there can be legal concerns whether it will be approved if it is alone AI discovered drug. And cost of AI implementation will add and may make the drug initially to some higher side. And data privacy and security, managing animal health data responsibility, and AI bias and reliability. Because the AI also makes some errors or bias, because whatever information has been fed by the human brain into the AI, it will make recommendations only up to that level. So ensuring unbiased AI-driven drug recommendation is a big challenge. And because this topic was very vast, I have restricted myself to drug discovery and delivery and up to veterinary sciences. So we can conclude that AI is revolutionizing drug discovery and delivery by making process faster, more efficient, and cost effective. AI-driven precision medicine is leading to personalized treatment for animals and as well as human beings. AI-powered drug screening, nanotechnology, and smart drug delivery are transforming veterinary medicine. Obviously, there are ethical considerations and regulatory challenges. They must be addressed for responsible AI adoption. And the future of AI in veterinary science is very promising. Sustainability, better treatments, and improved animal health care can be ensured with the help of these techniques. So thank you for your patient listening. If there are some questions, that can be.
Thank you, sir. Thank you so much for your excellent talk. Actually, you have explained all the things, the scope and the challenges and everything. The question we that has arisen in specifically my mind, you have addressed all of them. But still, we have some of the questions from the participants. So one question is, can AI make mistakes in analyzing and interpretation of data?
Definitely. That's why there are regulatory concerns, and it's not being. It's a new field. It's being tried, and whatever information or the informations we have fed into the system, it will analyze only up to that level. And the science is evolving, and so it can make mistakes. Definitely, it can make mistakes. It can make blunders, in fact. So the AI recommended diagnosis has to be counter-checked by the veterinarian or the medical doctor. And some approver has to be some human brain because it can make blunders, because it can go some erroneous side, some image recognition. It cannot be exactly same. It may sometimes I make my biometric information, my punch and face is being recognized by some other person, which may have some similar or errors. And I'm making my biometric presence, and some other guy is being made present. So out of 100, 1 or 2 errors are there. But this is not an error. This is a blunder, because I will be absent and he will be. So based on these, we cannot leave at this. This science is evolving. Once we are sure that up to what level of accuracy is there, only then we can, in a gradual stepwise process, we can release it to the patient. So that's the case.
Yes, sir, definitely. And we have received another question from Dr. Winkle. And the question is that, sir, since AI is a new technology, it would be costlier than how would it be applied at farmer level, making it cost effective practically?
Yeah, that's the one challenge, because I have discussed initially there may be some implementation costs, and the medication may be costly because the cost of development will go on onto the medicine. But in later stages, once these techniques are being globally used and these techniques are very widespread, the cost of medication will go down. So initially, and there may be cost of patents and all these things also. The cost factor is there, but in future, cost will definitely drop down, because the human interventions, the cost of medication, cost of development, if it comes down from 12 to 3 years, obviously the cost will go down. But all of a sudden, it cannot be. Once the infrastructure and all the logistics and support is fully developed, then it will. So that's the case.
Yes, sir. Sir, One another question that yeah, we our in our in other countries we don't know, but in about India, like in India, are there any institutes who are working in this AI based veterinary drug discovery, or this is still not going on in our country?
Uh, this AI is a new field and it's been taken up by so many institutes. In BSc also you will find that it is a Bachelor in Science in AI and all courses are coming up. But all the universities may not be having. I'm sure that some new courses, some diploma and other courses will come up in universities. And till now, it's only up to R&D level. Some pharmaceutical companies, they are making some in Aaj News channel, AI anchor is making news, but it's not universal in all the channels. So it's a new field. These programs will be implemented shortly in other universities also. But till now, in government universities in India, it's not there. It's coming up. It's a new field.
Yes, sir. Sir, as we know that technology is very good if we use it in a good way, but it's harmful also. It's dangerous also if we don't understand that what is the thin line between its pros and cons, and we need to use it wisely. So, on personal level, I would like to request you to address our participants and to give them a message to use AI wisely, to use it under your control, and don't get into it so much that you don't realize that where we have to stop. So I want to request you to give a message to the participants about its wise use.
Obviously, human brain cannot be replaced. We have to understand this. So many of our students, they are writing their thesis and their manuscript with the help of ChatGPT, and whatever it frames, it takes care of. But you are losing your writing skills. The, the person, if it comes to any platform, is not able to speak properly or is not able to draft any manuscript properly. And it's been we have to calculate up to what level we have to be dependent on these techniques. And why they have been developed? They have been developed to help us in treatment. So there's obviously there is a very delicate balance. Uses of, say, for example, uses of mobile phones and smartphones. We are becoming slaves of these machines, and very precious of our very precious time. Some 4 5 family members are there in a family, and each one is involved in some mobile or some WhatsApp chat or like that. So we have to decide their uses, and we have to control their uses also. And for veterinary or the medical field, suggestions or the recommendations or diagnosis coming from their side has to be counter verified, because there is legal ethical concerns are there. So just like use of phones and mobile phones and smartphones in our life, how we are controlling to what level I have to see, and thereafter I won't touch my phone if someone is making this. So accordingly, we have to adjust. These techniques are there. These techniques can destroy the career of any individual, or if it is leaving each and everything to the AI system, it may have some blunders, and the practice of the patient can be jeopardized. So those things has to be taken care of.
Yes, sir. I think there should be public awareness first about the harmful effects of this technology, because we came across a lot of news where people, they are conducting surgeries, watching YouTube videos and everything. So we should know that up to which extent we have to use this technology and at what level we have to use our brain. So that's why I just requested you to give a message to all the participants to wisely use the technology, so that we cannot get
There
harmed
are
with its
there are
effects.
AI-based platforms or online free platforms where you can submit your symptoms that I'm suffering from pain, fever, this. It will come up with suggestion, and if you start taking medicine, you may fall in big trouble. So we have to use our conscience where up to what level they can be used. If I have drafted a manuscript, if I want to check the level of plagiarism, I can take the help of AI. I can take the help of that's okay. But replacing my whole activity and surrendering to any AI or the computer system, definitely it will damage greatly.
Yes, sir. We should use it as a as help, not totally depend on it and not get into it so much that we forget to use our own brains.
Yeah, definitely.
Thank you, sir. It was indeed a very insightful talk. You have covered a very new topic, and I think most of the participants have understood your point of view, have understood the things which you wanted to convey. So, thank you so much, sir, for your
Thank
presentation,
you.
and we wish you all the best for coming future, and we hope that once again in some another training we came across and we discussed about some other topics and research. Thank you so much, sir.
Thank you. Thank you very much. Thank you.
Thank you,
Thank
sir.
you, sir.
Thank you, Dr. Dinesh and the whole team. Thanks for this platform. Thank you.
Thank you,
Thank
sir.
you.
Dear participants, it was a wonderful lecture from Dr. Nirbhay Kumar, and now moving forward, we will. I would like to invite our second guest speaker, which is Dr. Sitaram Gupta. Sir is a senior assistant professor and in charge Department of Veterinary Medicine, College of Veterinary and Animal Science, Rajasthan University of Veterinary and Animal Sciences, Bikaner. So Dr. Gupta holds a master's and PhD in veterinary medicine and has expertise and extensive professional and teaching experience. Sir has served in various academic positions, serving as assistant professor at Apollo College of Veterinary Medicine, Jaipur. Later on, he joined LRS Kodamdesar, Rajasthan University of Veterinary and Animal Sciences, Bikaner, and the Department of Veterinary Medicine at College of Veterinary and Animal Sciences, Bikaner. Dr. Gupta has achieved significant academic and professional milestones, including receiving prestigious awards like Veterinary Internal and Preventive Medicine Appreciation Award and the Best Clinical Award. Sir has published more than 70 research papers in various reputed national and international journals, along with 30 popular articles and clinical case reports. Additionally, Sir has delivered lectures in various training programs organized by State Veterinary University, Agriculture University, and ICR sponsored training programs for farmers and academicians. Sir is a life member of 10 professional society and journals and has also guided 6 MSc Masters of Veterinary Sciences students as a major advisor. So, sir, I must say we have to learn a lot from you, and we are very grateful to have you on board today. So, today, sir is going to talk on the topic as you can see on your screen. The topic is important diagnostic modalities in veterinary practice in reference to electrocardiography, ECG. So, sir, now this virtual stage is yours, and I am handing over the stage to you for your talk.
Thank you, Dr. Himani.
Over to you, sir.
Are you listening? Voice is clear?
Yes, sir. Voice is clear, but your camera is not properly adjusted. We can't. Yeah, you need to lower it down a little bit more. Yes. And also, you have to put your PPT on slideshow mode.
Yeah, I shared the slides.
Yes. But
If it is visible,
it's visible, but it's not
Camera
in
adjustment.
slideshow. Yes.
if it is same, but not visible.
So you can reshare it. You should. Yeah, stop sharing and then again share the PPT. I think it will work. Yes, it's visible. You can put it on slideshow mode so that participants can get it in a better way.
Now PPT is visible.
So we can see your screen, but it's like in the slides mode, not in presentation mode. So now it's visible.
Now it's visible.
Yes, now it's good. Perfect.
Let us start.
Yes, we can start.
Good evening, everyone. Today topic is important diagnostic modality in the veterinary practice in reference to electrocardiogram. As you know that the there are the various diagnostic modality which play an important role in veterinary practice as well as in the human practice also. Because as these diagnostic modality provide a very very important information about the internal organs, their functions, and so that the clinician make the proper decision, timely diagnosis, or can adopt the promote the good treatment, or this when the good treatment is achieved by the timely, then this important for the well-being of the animals. As you know, in present scenario, as you every know that the top most cause of the mortality in the human medicine is the cardiovascular disorder. This is the according to WHO, this is the top 5 causes in the cardiovascular diseases occur in the top 5 causes in the human mortality. In concern to veterinary medicine, the prevalence of the cardiovascular disorders are very low. This prevalence of the cardiovascular disorder, when we compared with the companion animal or large animal, the prevalence is very low in case of large domestic animal as compared to small animals, because the large animal in large animal practice only 10% of the animals are clinically examined by the clinicians for the cardiovascular disorders, because there is the lack of instruments which is giving the information of the cardiovascular disorders, or the large animal cannot be put for the cardiac workload, because the animal do not receive cardiac workload. So until unless the animal is in the terminal stage of the disease, cannot show the clinical signs of the cardiovascular disorder. So the animal is not diagnosed for the cardiovascular disorders. But particularly when we talk about the common cardiovascular disorder which is diagnosed in the large animal is the traumatic pericarditis. When we talk about the diagnostic mortality for the small animal, the prevalence of the cardiovascular disorders are large as compared to large animal, because they small animal or companion animals get the cardiac load when we take out from the house for walking, or then running, or then guarding, then they heart is load workload. So the animal shows the exercise intolerance, or there maybe shows the coughing. On that basis, the attendant or clinicians may suspect for the cardiovascular disorder. So in this today topic context, the these cardiovascular disorders can be diagnosed by the electrocardiography. So this is the very important diagnostic modality which is can be diagnosed the various cardiac disorder in the companion animal and the large animals. So now, when we talk about the electrocardiography, in this cardiography, we should know about the what are the electrocardiograph, what is the electrocardiogram, and what is the electrocardiography. Electrocardiograph, it is nothing. It is a machine, or it is a voltmeter, or it is a galvanometer, which measure the potential difference or changing the electrical activity of the heart between the positive or negative electron. And second is the electrocardiogram. It is a graphical representation of the electrical activity which is going on in the heart during the depolarization and repolarization. Means there are the graph is made in the graph is made on the ECG graph paper during the depolarization or repolarization. And last one is the electrocardiography. It is a process. It is a process by which electrocardiogram is prepared by the electrocardiogram. So this is the what are the utility and indication of the electrocardiography, because the cardiovascular disorders can be diagnosed by the physical examination of the animals, but the possibility of the cardiovascular disorder cannot diagnosed by the physical examination. So the electrocardiography, which tells the various cardiovascular disorder which cannot be diagnosed by the physical examination, and this is the very easy to examine the animal. This is the non-invasive and quick diagnostic tools, and this electrocardiography easy to perform and easy to read, and results are immediately available. In other diagnostic modalities, there are required sometimes to achieve the results of the overdiagnosed. We receive the results after some time. But in ECG, we can get the results immediately. And this ECG may also be provide the additional information to facilitate the diagnosis and treatment of the cardiovascular diseases. The ECG may also be helpful in evaluation of the animal for the in case of acute emergency cases, or in case of cardiac compromised animal can be easily or better way diagnosed by the electrocardiographic examination. There may be different type of arrhythmias may also be evaluated by the electrocardiographic examination. If there may be any cardiomegaly or any enlargement which enlargement present in the heart may also be diagnosed by the electrocardiographic examination, because in case of X-ray examination we can be diagnosed the cardiac enlargement or hypertrophy, but which part of the heart is showing the enlargement or hypertrophy cannot be diagnosed by the radiograph, but the electrocardiography will make that diagnosis for which part of the heart is involved or showing the sign of showing the hypertrophy or enlargement. And evaluation of the PET for the pericardial effusion may also be done by the electrocardiography, and cardiac evaluation of the disease associated with the multiple organ failure, because sometimes the ECG, if there is the renal failure, then this renal failure may also be change the electrolyte. So there is the difference in the different waves may also be diagnosed by the electrocardiography. Now, what are the requirements for the electrocardiography? There is no special requirement for the cardiographic examination, only required for the one ECG machine along with the leads. And third requirement is the ECG gel or spirit which prevent or which remove the air between the hairs. And fourth one is the ECG paper, and last one is the cotton rolls. Now in lead system, how to get the ECG in the animal? In animal, there is the 6 lead system is most commonly used. In this 6 lead system, one is the bipolar standard lead system, and second one is the augmented unipolar limb lead system. This is the bipolar lead system. This is the 3 lead 1, lead 2nd, and lead 3rd. And lead 1 is placed at the right arm and compared with the left arm, and lead 2nd is placed between the right arm and left leg, and lead 3rd placed between the left arm and the left leg. These leads in bipolar standard lead system, the lead is a combination of the 2 leads which compare the electrical potential of the 2 different electrode. 2 different electrode means in lead first, the first electrode, which is negative electrode, is placed on the right arm and positive electrode present on the left arm. In lead 2nd, the electrode is placed, negative electrode is placed on the right arm and the positive electrode is placed on the left leg. In lead 3rd, the negative electrode is placed on the left arm and positive electrode placed on the left leg. And in this bipolar standard lead system, the right hind limb is act as a neutral. And in augmented lead system, this augmented lead system, this compare or this measure the electrical potential of the one positive electrode and compare with the 2 other electrodes. In this system, the lead is the AVR and lead AVL and lead ABF. And AVR stands for the augmented vector right, and AVL stands for the augmented vector left, and ABF stands for the augmented vector foot. In this system, the right arm acts as a positive electrode, and remaining left arm and left leg act as a negative electrode. And this achieve the potential difference between the right arm and average of the left arm and left leg. And similar the ABR, the lead ABL give the electrical difference between the left arm and average of the right arm and left leg. And similar in ABF, the potential difference measured by the left leg and average of the right arm and left arm. The preparation of the animal for the ECG. There is no special preparation is required for recording of the ECG. In recording of the ECG, first important requirement is the animal position in the right lateral recumbency. And second important preparation is the animal should not be restrained by the chemical mode. So means there is no requirement of any sedative, any tranquilizer for restraining of the animal. And third requirement for taking the ECG is there is no metallic chains or collars should be present on the animal body. And next one precautions is there is no direct contact of the animal body with the electrocardiographic tables, because if the table and the other metallic items which is present on the animal body, then this disturb the electrical activity which is going on the heart. So these precautions should be kept in mind while we prepare the animal for the ECG. Now, after preparation of the animal for the ECG, then placement of the leads. As discussed in the earlier slide, there is the 6 lead, which is three leads are the bipolar standard lead, and 1 is the augmented unipolar lead. These leads are placed on the flexor angle of the elbow in the forelimb, and the flexor angle of the staple joint and the hook joint in the hind limb. This is the pictorial presentation by this shows the placement of the limb leads in the hind limb. This is the flexor angle of the hook joint, and in this slide, this is the flexor angle of the elbow, just caudal or just dorsal to elbow. And before placing the lead on the limbs, we should apply the spirit or gel to remove the air trapped between the hairs. Now this is the normal conduction system, and this slide tell us that from electrical impulse generate and how this electrical impulse spread all over the cardiac region. Then what event should be recorded in the ECG paper during the transmission of these electrical impulse from the atria to ventricle. In this conduction system, the normal conduction system is electrical impulse is generated from the SA node, and this SA node acts as a pacemaker for the heart. From this SA node, the electrical impulse which is generated is spread all over the atria, and after that, this electrical impulse reaches to AB node by the internodal pathway. From the AB node, this electrical impulse goes to the ventricle by the bundle of his and left bundle branch block and right bundle branch block, and lastly the Purkinje fibers. And during the transmission of electrical impulse, there are the various sequence of event are happen, and these sequence are happen with the constant interval. When there is the impulse is generated from the SA node and is spread in the atria, and this leads to atrial contraction, and after atrial contraction, this conduction goes to AB node and spread in the ventricle by the bundle of his and left bundle branch block and left right bundle branch and Purkinje fiber, and this leads to ventricular depolarization and ventricular contraction. And this slide shows the different activity which are traced on the electrocardiographic paper during the normal conduction system, and by the different waves, segments and interval are traced in the ECG paper. And in this ECG paper, we measure the 5 waves, 2 segments and 2 intervals. When the electrical impulse generated from the SA node and spread in the atria, then cause the atrial depolarization. In this atrial depolarization is found in the form of P wave in the ECG paper. And after complete depolarization of the atria, then this electrical impulse goes to AB node, and from AB node, this electrical impulse goes to bundle of his and left bundle branch or right bundle branch, and this is form a QRS complex in the ECG paper. And after that, there is the repolarization of the heart is found in the form of T wave in the ECG paper. And there are the 2 interval, this PR interval, which includes the impulse on the electrical activity which occur in the atrium. And this QT interval provides the information about the electrical events which is happen in the ventricle. Now, this is the ECG paper. In this ECG paper, there are the several large boxes, and these one large boxes has 5 by 5 millimeter of small boxes. So we should know about the what are the dimension in the horizontal line and the vertical line. In horizontal line, we provide the information of the duration, and the vertical line will provide the amplitude or voltage of the amplitude, electrical impulse. So in standard ECG paper, the sensitization or speed of the ECG paper is 25 millimeter per second, and the voltage of standard ECG paper is the 10 millimeter per centimeter. So in this regard, this is the 5 millimeter wide, and height of this box is the 5 millimeter. At the speed of 25 millimeter per second, each small box is formed by the 0.04 second in the horizontal line and 0.1 millivolt in the vertical line at the voltage of the 10 millimeter per second. So this is the very important, because the coming slide we measured the P wave duration, PR duration on that data. So it is clear to all the participants is the one small box is defined by the 0.04 second and 0.1 millivolt. This is the normal electrocardiographic parameter in the healthy dog. In this slide, the normal P wave duration is the in the 2 small box, and height of the P wave is the 4 small box. This 2 small box means indicate the P duration is normal up to 0.08 second, and amplitude of the P wave is normal up to 4 small box means 4 millivolt, and the PR interval is normal in the up to 2 to 5 small box. 2 to 5 small box means there is the PR interval is normal up to 1 large box, and 1 large box is indicate the 0.2 seconds, and QRS complex is normal up to 3 small box, and amplitude of the R wave is normal up to 2.5 millivolt to 3 volt, and the T wave amplitude is the 1/4 of the R wave amplitude, and the duration of the T wave is normal up to 2 small box. So this is the normal parameter by which ECG is present, and by comparing these normal parameter by the diseased animals, we can diagnose the animal is suffering from the atrial enlargement, atrial hypertrophy, or ventricular enlargement or ventricular hypertrophy. So what information obtained from the ECG? While after getting the ECG from the animal, following information can be obtained from the ECG. First one is the calculation of the heart rate, determination of the heart rhythm, complex measurement means there is the measurement of the duration of the different waves and segment, and amplitude of the different waves, and calculation of the mean electrical axis and type of cardiac arrhythmias. So how to calculate the heart rate? In case of regular rhythm, there is the method of large boxes method or small boxes method. If the rhythm is irregular, then heart rate may be calculated by the 3-second method or 6-second method. In regular rhythm, heart rate is calculated by the large box or small box. The formula for calculation of the heart rate in the regular rhythm is the 300 divided by number of large box between the two R waves. And when the speed is 25 millimeter per second, then 300 divided by number of large boxes between two R wave, or 1500 divided by number of small boxes between two R waves. This is the same way, because we are in one small box. There is the one large box contain the 5 small box. So this 300 divided by number of large box between the two R wave is same as the 500 divided by number of small boxes between the two R wave. And if the speed of the ECG is the 50 millimeter per second, then this formula is converted by the 3000 divided by number of small boxes between the two R waves. When the rhythm is irregular, then we use the 3 second or 6 second method. 3 second 35 millimeter per second speed, then we calculate the number of QRS complex in the 3 second, and number of QRS complex in the 3 second is multiplied by the 20. And if we calculate the heart rate by the 6 second method, then number of QRS complex in the 6 second and multiply by the 3rd second. Now we calculate the heart rate in these slide, these ECG. In this slide, there is the number of large boxes between the two R wave. This is the one R wave. This is the second R wave. We calculate the number of large boxes between these two R waves and this number of large boxes, and divide the 300 by the number of large boxes between the two R waves. Now calculate 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Means there is the 10 large boxes present between the two R waves. So now how to calculate the heart rate? In this is the rhythm is regular. So number 300 is divided by number of large boxes between the two R waves. When in this example, the number of large boxes is the 10. So heart rate is the 30 divided by 10 is the 30 heart beat per minute. And this is the sinus bradycardia. In this slide, the number of large boxes between the two R wave is the 1 and 1 large boxes and 2 small boxes. Means there is the 5 or 2 7 small boxes. So heart rate is the 300 divided by 7, almost 70 or 75 heartbeat per minute. So in tachycardia, the heart rate is the 200, because when we calculate from by the small boxes, then this number of 500 is divided by the number of small boxes. If we calculate the large boxes, then 300 is divided by the number of large boxes between the two R waves. So in this lower ECG graph paper, there is the heart rate is the 150, approximately 150. So this is the sinus tachycardia. Now there is the abnormality in the P wave. As discussed in the previous slide, the P wave, the duration of the P wave is normal up to 2 small box. If the duration of the P wave is more than 2 box, then this is due to left atrial enlargement of P pulmonale. If the amplitude of the P wave is more than 4 small box, this indicates the right atrial enlargement. And if there is the P wave is tall and broad, in that condition, the condition indicates the bilateral and atrial enlargement. If there is the variation in the amplitude of the P wave, this indicates the wandering pacemaker. And if there is the absence of P wave, that this indicates the atrial stand-still, silent atrium, or sinus block. This is the ECG finding of the P wave. In this first picture, the duration is the up to 2 small box. So this is the normal, and amplitude is up to 4 small box is not normal. So in first ECG, the duration is the 0.4 and amplitude is the 0.3 is the normal. If in the tall P wave, the amplitude of the P wave is more than 4 small box, and in this ECG, this the amplitude is the 0.5 millivolt. So this is the tall P wave. This indicates the right atrial enlargement. In this ECG, there is the duration of the P wave is more than 2 small box, and amplitude is the up to 3 small box, means the amplitude is normal and the duration is the enlarged. So this indicates the left atrial enlargement. And there is the abnormal waveform in the QRS complex. The abnormal QRS complex may provide the information about the ventricular hypertrophy, ventricular enlargement, or bundle branch block, either left bundle branch block or right bundle branch block. If the amplitude of the R wave is more than 2.5 or 3 millivolt, then this indicates the left ventricular enlargement of or left bundle branch block. If the duration of the QRS complex, as discussed in the earlier slide, the up to 3 small box is the normal. If the QRS complex is more than 3 small box, then this indicates the left bundle branch block or left ventricular enlargement. And if there is the deep S wave or deep Q wave, deep S wave and deep Q wave indicate the right ventricular enlargement or right bundle branch block. And if there is the abnormality in the T wave, this indicates the electrolyte imbalance. So apart from the cardiovascular disorders, ECG can also be provide the imbalance of the electrolytes. And this is the very important is the ST segment, because this ST segment provides the information about the myocardial infarction, myocardial ischemia. There is the hyperkalemia, hypokalemia, or any kind of pericarditis. So this is the very important. These ST segment are the isoelectric line. If there is the depression or elevation, this provide the information about the ischemia and infarction in the heart. So these are the various examples of the atrial enlargement. In this example, B shows that there is the increased amplitude of the P wave. This increased amplitude of the P wave indicates the right atrial enlargement. In below this ECG, there is the widening of the P wave. This widening of the P wave indicates the left atrial enlargement. This is the deep S wave. This is the Q wave. After Q wave, this is positive deflection is the R wave, and after positive deflection of the R wave, this is the first negative deflection is the S wave. In normal ECG, this is the deflection of the S wave is only up to 2 small box, but in this case, the S wave is the very deep, and this deep S wave is indication for the right ventricular enlargement. And similarly, in this ECG, the deep Q wave, this is the, in normal ECG, the Q wave is first negative deflection, which is up to 1 small box. But in case of right ventricular enlargement, this Q wave goes down up to 3 or 4 small box. So there is the deep S wave or deep Q wave indicate the right ventricular enlargement. So next information we can get from the electrocardiography is the mean electrical axis. This mean electrical axis, it measures the overall electric direction of the electric current in the heart. So in determination of the mean electrical axis, make a circle by this electrical axis is measured on the frontal plane of the ECG leads, and this the normal mean electrical axis is the 40 to 100 in the case of dog. In case of cat, it is normal up to 0 to 160 degree for the cat. But when there is the mean electrical axis between the 40 to -90, this indicates the left axial deviation. If the mean electrical axis more than 100 degree to -90 degree, this indicates the right axis deviation. This axis deviation occur due to there is the any infarction, any ischemia, any hypertrophy of the any part of the heart, or any enlargement, or any bundle branch block. So to determination of the axis, mean electrical axis, we observe the QRS complex. In QRS complex, we observe the lead first and lead AVF. The QRS complex of the lead first and lead AVF will be observed to determination of the mean electrical axis. In these three electrocardiographic observations, I explained how to calculate the mean electrical axis. In the first example, there is in lead first, the QRS complex is the positive deflection, and in ABF, this is also a positive deflection. If lead 1 and lead AVF shows the positive deflection, this indicates the normal axis. And there is the one mnemonic for the determination of the mean electrical axis is the right reaches and left leaves. Means in this second electrocardiographic report, in the lead first, the QRS complex is positive deflection, but in case of ABF, the QRS is the negative. Means both QRS complex never meet to each other. So these are separating to each other. So this is the left axis deviation. It is clear. And in next ECG report, the QRS complex in lead first is the negative deflection, and ABF, this is the positive deflection. And in the both the leads, the QRS complex are meet. So this is the right axis deviation. This is the simplest method by mean electrical axis determination. There are the various methods, but this is the simplest method by which we can determine the mean electrical axis of the heart. And now is the arrhythmia. Before going to arrhythmia, we should know about the rhythm. Rhythm it is a sequence of the event which occur during the conduction of the heart. If there is the disturbance in the conduction of the heart in the form of rate, regularity, or site of origin, then this is called as arrhythmias. On the basis of disturbance, if there is the disturbance in the speed, or there is the disturbance in the site of origin, or there may be disturbance in the conduction system, on that basis the arrhythmias may be classified as the disturbance in the impulse formation and disturbance in the origin and disturbance in the impulse conduction. So first class in classification of the arrhythmias, there is the normal sinus impulse formation, means normal sinus rhythm. Normal sinus rhythm means the electrical impulse is generated from the SA node and goes to AB node by the internodal pathway and reaches to bundle of his left bundle branch block and right bundle branch block and Purkinje fiber. If the conduction occur in this sequence, this is the normal sinus rhythm. If there is the disturbance in the speed, but the conduction is normal, but there is the difference between the speed, on that basis the arrhythmia is the sinus bradycardia or sinus tachycardia. And if there is the disturbance in the origin, as discussed, the impulse is generated from the SA node and trouble AB node by the internodal pathway and goes to bundle of his left bundle branch block, left bundle branch, right bundle branch, and Purkinje fibers. If the impulse generate other than the SA node but above the ventricle, and this type of arrhythmia is known as supraventricular impulse formation, because the impulse is generate above the ventricle. So that's why this type of arrhythmia is known as supraventricular arrhythmias. And this supraventricular arrhythmias, there are the various type of arrhythmias. First one is the atrial premature complex, atrial tachycardia, atrial fibrillation, atrial flutter. If the origin of the impulse is from the below the atria or from the ventricle, then this type of impulse arrhythmia is known as ventricular impulse formation and ventricular rhythm. And there in this category we are discussed the ventricular premature complex, ventricular tachycardia, and ventricular fibrillation. If there is the disturbance in the conduction, on that basis there are the cardiac arrhythmias is the first degree AB block, second degree AB blocks, third degree AB blocks, and left bundle branch block and right bundle branch block. These type of arrhythmias is due to there is the disturbance in the impulse conduction. Now, and coming slide is giving the information about the different kind of arrhythmias, and all these different kind of arrhythmias interpreted by these 6-step method of the interpretation of the ECG. In first step, we calculate the heart rate, and this is the normal or abnormal. And after calculation of the heart rate, we see the ECG for the regularity. It is the ECG regular or irregular, and after that we see the ECG for the P wave. If the P wave is present or absent, if there is the present, then this P wave is normal or abnormal. After that, we calculate the PR interval, and this PR interval is constant or variable, or there may be present or absent. This PR interval absent when there is the no P wave in ECG. And after calculating the PR interval, we examine the QRS complex, and in QRS complex, we measure the duration of the QRS complex and the morphology of the QRS complex. And after calculating the heart rate, regularity, P wave, PR interval, and QRS complex, after that, we interpreted the ECG. On that basis, the ECG is sinus, rhythm, or arrhythmias. So first is the sinus rhythm. Sinus rhythm means each and every electrical impulse is generated from the SA node, and SA node to AB node, bundle of His, and left bundle branch, right bundle branch, and Purkinje fiber. And how to diagnose the ECG? This is the sinus or not sinus. In sinus rhythm, each P wave is married to the QRS complex, and after the QRS complex, each beat has the T wave. If there is any abnormality in the PQRS complex and T wave, then this is the sinus arrhythm. If these present in the each beat, then this is the sinus rhythm. In this ECG, we see the P wave, and this is followed by the QRS complex, and this is the inverted T wave. And after that, this is the P wave, and this QRS complex and inverted T wave. And this is P wave, inverted QRS complex, and inverted T wave. Means there is each and every P wave is originated from the SA node, and after that there is the QRS complex, means there is the ventricular depolarization, and T wave, this is the ventricular repolarization. And there is between two R wave, the number of large boxes is the same. Means this indicates the regularity of the rhythm. And calculate the heart rate. In this ECG, the number of large boxes between the two R waves is the 3, 1, 2, and 3. Now heart rate is the 300 divided by 3, means 100 heartbeat per minute. This is the normal sinus rhythm. In case of sinus bradycardia, means all the electrical impulse generated from the SA node, but the duration is the slow. So the gap between the two R wave is increased. So in this slide, we can say that number of R wave between the two large box between the two R wave. This is the 1, 2, 3, 4, 5, 6, and this is the half in this side. So almost 7 large boxes between the two R wave. So this is heart rate is the 300 divided by 7, approximately 40. So this is the bradycardia, and regularity is normal or regular because the between two R wave, the number of large boxes is the same, and the P wave is present, and P wave is followed by QRS complex, and after that this is the T wave. So this is the sinus rhythm. In case of tachycardia, the number of large boxes between two R wave is less. So in this ECG, we calculate the number of large boxes between two R wave is the almost 2. So heart rate is 150, approximately 150, and each P wave is married to QRS complex, and after that there is the T wave. So this is the sinus tachycardia. In case of atrial premature complex, this is the supraventricular impulse formation, because in this cardiac arrhythmias, there is some ectopic foci which may also be present in the atrium, and apart from the SA node, sometimes this ectopic foci may control the they may discharge the electric impulse and counter the acid node impulse. So in ECG, there may be some different morphological P wave is present. So if when the pattern of the different morphological P wave presents in the ECG, on that basis, the APC may be divided into biseminal APC and triseminal APC and quadriseminal APC. And now explain this ECG. In biseminal APC, in this APC, this is the P wave. This is followed by the QRS complex and T wave. But there is the difference in the morphology of this P wave. And this is the normal P wave. This is the different morphology of the P wave. So this different morphology of the P wave present in the ECG, this indicates the APC. If this APC present every second beat, when this APC present at the every second beat, then this called as biseminal APC. If these APC present every third beat, then this is called as triseminal APC. And if the APC present every fourth beat, then this APC is known as quadriseminal APC. So this is the different morphology and increase PR interval. This is the important finding which we seen in the ECG while diagnosing the APC. In the triseminal APC, every third heartbeat is APC. This is the first normal sinus rhythm, and this is the normal sinus rhythm. This is the APC, because in third beat, there is the morphology of the P wave is the different. And similarly, this rhythm is normal, and this is the normal, and this is the different morphology, and this is the EPC. Means this is the every third heartbeat is originate prematurely by the ectopic foci. In this, the quadriseminal APC means every fourth heartbeat is originate from the ectopic foci. So this is the first normal P wave. This is the normal P wave. This is the normal P wave. This is the inverted P wave. This is the different morphology. Is the fourth beat. So this is the quadriseminal APC. And now is the atrial flutter, and discuss in the atrial flutter or atrial fibrillation. This both are due to there is the presence of the ectopic foci in the atrium. Apart from the acid node, these ectopic foci also generate the electrical impulse and produce the wave in the ECG. In atrial flutter, it is the rapid regular atrial tachycardia. This atrial tachycardia is not due to firing of the acid node, but it is due to ectopic foci. In this atrial flutter, the heart rate is more than 300 to 400 per minute. And in this atrial flutter, the ectopic foci is the large and in the single. But due to this large single ectopic foci generate the electrical impulse, and this electrical impulse is spread all over the atria, and again this electrical impulse is enter into ectopic foci, and due to this the electrical current is generated repeatedly. So this phenomena is known as circus re-entry phenomena. By this, there is the several P wave is formed between the two QRS complex, and this flutter wave in ECG paper shows the certain ratio or certain pattern, like as 2:1, 3:1, and 4:1. This 2:1, 3:1, and 4:1 indicate the two flutter wave is followed by the QRS complex, and 3 atrial flutter followed by the 1 QRS complex, and this 4 flutter wave followed by the 1 QRS complex. In this atrial flutter, there is the no formation of the P wave, because the P wave is not formed, because these waves are formed by the ectopic foci, not a SA node. Now, this is the ECG of the atrial flutter. In this, these are the shows the like a sawtooth appearance, just like a sawtooth. These sawtooth appearance is the flutter waves, not a P wave. And after that, this is the QRS complex. When this QRS complex is formed when the electrical impulse comes from the SA node to AB node, and this type of pattern which is present in the ECGs, sawtooth pattern, is indication of the atrial flutter. And for diagnosing the atrial flutter, we see the electrocardiographic paper by the reverting the image, just like this. This is also is the atrial flutter and showing the sawtooth appearance, but due to presence of QRS complex, there is the no much difference. But when reverse the ECG, then this give the completely sawtooth appearance, and this is the atrial flutter. Now, atrial fibrillation, just like a atrial flutter, there is so many ectopic foci which is present in the atrium, and each ectopic foci generate the electrical impulse, or these electrical impulse causing the very fast or irregular atrial contraction. Due to very fast irregular atrial contraction, causes the not synchronize the each other. Due to not synchronization of the atrial contraction, does not generate the pulse. When we examine the patient by the pulse examination, then the patient shows the normal pulse. But when we put the stethoscope on the chest region or on the cardiac region, we measure the heart rate is more than 300 to 600 beat per minute. So this difference is known as the pulse deficit. And this pulse deficit is clinically examined when the animal is examined by the physical examination. And in this atrial fibrillation, the P wave is absent, but these presence of the fibrillary wave, in just like a atrial flutter, in atrial flutter, the flutter wave is present. In the atrial flutter, the waves are denoted by the capital F waves, and in case of atrial fibrillation, the waves are denoted by the small p waves. And this is the electrocardiographic, which shows the atrial flutter, atrial fibrillation. And this is the irregular rhythm. In the between the two R wave, there is the almost 2.5 large boxes. But in case of this this between the two R wave, this is the 1.5 large boxes, and this between that place, there is the almost three large boxes. So this is the regularities irregular, and there is the number of fibrillary wave is present, and otherwise all the QRS complex is normal, and the duration of the QRS complex is normal, but the PR interval is absent because there is the P wave is absent. So this is the very important ECG finding for the atrial flutter and atrial fibrillation. There is the no PR interval, because in both the condition the P waves are absent. Instead of the P wave, there is the fibrillary waves are present or flutter wave are present. So the interpretation of this ECG is the atrial fibrillation. This is the electrocardiographic record of the atrial fibrillation and atrial flutter. Now this is the ventricular premature complex. In ventricular premature complex, the impulse is generated from the ectopic foci which is present on the ventricle. In atrial premature complex, the electrical impulse is generated from the ectopic foci which is present on the atrium. And in this BPC, the electrical impulse is generated from the ectopic foci which is present on the ventricle. So when the electrical impulse which is generated from the ventricle which not pass from the AV node, so in this condition there is the QRS complex is the wide and bizarre. So this is the important ECG finding when we diagnose the ventricular premature complex. In ventricular premature complex, there is the normal sinus rhythm which is interrupted by the one ectopic electrical impulse which is generated by the ectopic foci. So this is the ECG of BPC, and in this electrocardiographic report, this is the normal sinus rhythm, and this normal sinus rhythm in this after that this is the BPC, because this QRS complex is formed by due to electrical impulse which is generated by the ventricular ectopic foci. This is the normal sinus rhythm, and after that this is the BPC. If so, this is the BPC, and now ventricular tachycardia. This is the similar as the BPC, but in this ventricular tachycardia, there may be presence of the 3 or more BPC in the continuously in the ECG. If there is the more than 3 BPC is present continuously, then the electrocardiographic diagnosis is the ventricular tachycardia. If the less than 3 BPC is present in the continuously, then the condition is known as ventricular premature complex. In this ECG, there is the continuous more than 4 BPC is present, and the morphology or QR complex is normal, because when the impulse generated from the SA node, and when this impulse electrical impulse goes from the AB node, then there is the narrow sharp angle and steep QRS complex is formed. But when the electrical impulse generated from the ventricles, then the morphology of QRS complex is wide and bizarre shape. So this is the ventricular tachycardia. In case of ventricular fibrillation, just like a atrial fibrillation, in ventricular fibrillation, there is the several ectopic foci which is present in the ventricle, and these several ectopic foci which is present in the ventricle discharges or generate the electrical impulse. On that condition, the SA node which is controlling or pacemaker of the heart is not working properly, and these ectopic foci take the charge of SA node. In that condition, there is the no rhythm. The rhythm is nonsense. There is no fixed pattern of the P wave QRS complex, and this type of ECG is present when there is the ventricular fibrillation. The regularity is the irregular, P wave is absent, PR interval is absent, QR complex is absent, and in there is no sense of the any kind of rhythm. And the last type of cardiac arrhythmia is the AV block, and this AV block is the delayed conduction of the impulse through the AV node or bundle of His. The different pattern of the delayed in conduction, on that basis AV block may be classified as the first degree heart block or second degree heart block or third degree heart block. In case of first degree heart block, all the atrial conduction goes to ventricle, but in delayed condition. Means all the electrical impulse goes from SA node to AB node or AB node to bundle of SA and bundle branch and Purkinje fiber, but the duration from the AB node impulse is delayed. So when there is a delayed in the electrical impulse from the AB node, this denoted by the prolonged or constant PR interval. So in case of first degree heart block, in ECG, all the wave or segment and intervals are present, but the interval of the PR interval is the prolonged. But in each beat, the PR interval is constant. So in this slide, we see the P wave, and this is the QRS complex, and this is the T wave, again P wave, QRS complex, and T wave, P wave, QRS complex, and T wave. And when we measure the PR interval, then in that condition, in this ECG recording, the PR interval is more than 2 large box and almost 3 small box. But the normal PR interval is up to 5 small box or up to 1 large box. And the similar PR interval is constant and enlarged. So this is the sinus bradycardia or first degree heart block. In this condition, if we calculate the heart rate, then the number of large boxes between 2 R wave is 1, 2, 3, 4, 5, and almost 6 large boxes present between 2 R waves. So heart rate is 60. This is the less than the normal. So this is the bradycardia, and this is the first degree heart block. In case of second degree heart block, some atrial conduction goes to ventricle, and some electrical conduction does not reach to ventricle. So in ECG, some P waves does not have the QRS complex, and some beats has the proper sinus rhythm. So in that condition, on that basis, second degree AV block may be divided into 2 types. In first one is the type 1 second degree heart block. This is also known as Banky-Beck heart block. In this heart block, the PR interval is progressively increased, and this progressively increase of PR interval up to the P wave is not conducted. Means the PR interval is increased, means that there is the delayed conduction, and after sometimes there is the no conduction, and when there is the no conduction, then there is the no presence of the QRS complex. And so see this electrocardiographic findings. This is the P wave and QRS complex. This means this is the normal conduction of the normal atrial conduction. This T wave, this P wave, and this QRS complex, this T wave, this P wave, QRS complex, and T wave, and this P wave, but there is the absence of the QRS complex. So, but some beats are conducted and some beats are not conducted. When the beats which are not conducted towards the ventricle, then there is the absence of the QRS complex. And in where the conduction occurs, but there is the increased PR interval continuously. That seen in this place, there is the PR interval is 11 millimeter, and in that place this is the 16 millimeter, and this is the 18 millimeter. And after there is the no conduction of the P wave, so there is the absence of the QRS complex. In case of Mobis 2 type of second degree heart block, and there is the intermittent block of the QRS complex, or there is the intermittent block of the P wave. So in this, there is the constant increased PR interval followed by the absence of the QRS complex. So in this ECG, this is the P wave, this is the QRS complex, and the morphology is changed. In this second type heart block, the QRS complex is wide or bizarre shape. And after that, this is the T wave, and again this is the P wave, and this again is the QRS complex, and P wave, and there is the absence of the Q waves. And again, this is the P wave and QRS complex. So in second degree, second type of second degree heart block, the PR interval enlarged and constant, but there is the intermittent P wave block. In case of third degree or complete AV block or complete heart block, means there is the no impulse is transmitted from the atria to ventricle. So this is the very fatal heart block. In this heart block, there is the no any relation between the P waves and QRS complex. In this condition, the atria and ventricle are contract independently. So in this ECG finding, there is the wide QRS complex, but there is the absence of the P wave, and QRS complex is present. And there is the presence of the P wave, but there is the absence of the QRS complex. So there is in this third degree of heart block, there is the number of P wave is more than the QRS complex. And in this third degree heart block, the duration of the QRS complex is more than 3 small blocks, means there is the more than 0.12 second. So this is the third degree heart block. And this is the we conclude the this topic by the ECG finding. We can be diagnosed the various cardiovascular disorder that cannot be diagnosed by the physical examination of the animals, like enlargement, hypertrophy, bundle branch block, and different kind of arrhythmias.
Thank you, sir. Thank you for the detailed presentation, and I'm sure that participants must have gained most of the knowledge from your presentation. So we have some questions from the participants. So one question is, how expensive is the ECG machine, and are there any brands you can suggest and what is the minimum investment we need to start ECG at clinic?
Ma'am, nowadays there is the various diagnostic modalities are available. So when we are diagnosing the cardiac diseases, then this ECG is very important. And in the concern of the rate, this is the not so much costly, and this can be getting from the 20,000 to 1 lakhs. The minimum, this is the depend on the version. And if we taking the high end version, then this is the very costly. If we getting the normal version of the ECG, then this is the not much so costly, up to 20,000, 25,000 can we get.
Okay, sir. One another question we have got, that sometimes machine deflection may be wrong. So how to identify and interpret it?
This is a good question. All the conditions which is occur in the cardiovascular disorder can be diagnosed by the electrocardiographic examination. For example, if there is I discussed as there is can be diagnosed by the cardiac enlargement and hypertrophy by the ECG, but cannot be measured how much thickness is increased in the hypertrophy and enlargement. So in that condition, we should adopt the echocardiography. When the information we cannot get from the ECG, then all these information we can get from the echocardiography.
Okay, sir. Thank you, sir. And I can see in the chat box, participants have learned a lot from your presentation, and they are requesting again and again to share the presentation with them. So you can share the presentation with Dr. Dinesh so that we can share it with the participants. It will be very, very useful to them.
Share the presentation with the doctor.
Thank you, sir.
Thank you, sir.
So much.
Your very informative and nice presentation. Really, all participants enjoy this innovative whatsoever you learn or did in the clinics that you show to the all participants. So it is very informative. So thank you, sir.
Thank you,
for inviting for accepting my invitation.
Very large presentation. This presentation, there is more than 50 slides. So I
Yes.
concluded speedily so that the completed the timely this presentation.
Yes, sir, but
Okay,
it was
okay, sir.
an important topic.
I
But
also share
this
your contact
presentation,
number to participants, sir. If anyone want to know more, then you can directly interact with them, sir.
okay, okay, sir. Sir, I
Definitely, sir.
just
Thank you so much, sir.
thank
Thank
you,
you,
sir.
sir.
Thank you,
Thank
sir.
you for this detailed presentations and giving your precious time to us and our participants.
Thank you, ma'am, and thank you, organizers for giving this opportunity. Thank you.
So,
Thank
dear
Thank
you,
you so
sir.
much, sir.
participants, yeah, dear participants, as we have reached to the end of today's session, so on the behalf of the organizers, which are Biotech Agriculture Forum for Technical Education of Farming Society, Kota, Faculty of Veterinary Science and Animal Husbandry, Ranchi, Birsa Agriculture University, Ranchi, and the entire organizing team, I deliver my heartfelt gratitude to our amazing speaker, Dr. Nirdha Kumar and Dr. Sitaram Gupta, sir, for their insightful talk. They have well explained their topics and satisfied your queries and given their precious time to all of us. So we are very, very thankful to our speakers, and I'm very sure that their hard work and experience must have motivated you all and must have boosted your knowledge and inspired you to work so hard in your field so that you can be on such a platform that they are today. So thank you, and we wish all the best to our speakers and participants for their future after this learning. And lastly, I would like to thank the participants for joining this 7 days training program and for being so patient and attentive throughout today's session and maintaining the decorum of the training. So thank you all and have a great evening. Thank
Thank
you,
you.
everybody.
Thank you, everyone. See you tomorrow again.
Yeah,
Thank
see
you.
you tomorrow, everybody.